Welcome back. We're going to talk about protein synthesis now. So DNA, which is found in the nucleus, serves as the blueprints for making proteins. DNA never leaves the nucleus, but it codes for RNA, or it's used to make RNA, which are the workhorses of protein synthesis. Within DNA, we have regions called genes, and genes are the segments that carry the information for a specific proteins. And if you remember from chapter 2, we talked about proteins being either fibrous or globular. Fibrous proteins tend to be more structural, so they're going to be for strength. They give form, whereas globular proteins are going to have multiple functions. An example would be an enzyme. So DNA codes for, is coded in a series of bases. And so for every 3 nucleotides in DNA, we have 1 amino acid being coded for. So over here you can see an amino acid codon table. This is an RNA codon table. So you can see these are all RNA nucleotides. So uracil, cytosine, adenine, and guanine. And within this, you have every single amino acid. So we have 20 amino acids total, but there are 64 codes, 61 codes code for amino acids, and then 3 code for stop. Right here, here, and here, okay? So I'll come back to this table when we're talking about translation and how we translate or produce the protein itself. Protein synthesis itself occurs on ribosomes, which are found in the cytoplasm, mainly on the endoplasmic reticulum, but also free-floating. DNA doesn't leave the cell. It's kind of like the CEO of a company. The CEO tends to stay in his office. He's not seen as often. So I'm saying this, that may not be true because I don't know many CEOs. But when you think about it, this is my best explanation. So the CEO, the person in charge, may sit in his office, but then they talk to the foreman, and the foreman is the one that comes out and actually tells the workers what to do. So DNA doesn't leave the cell. Instead, we produce RNA from the DNA. And the RNA is like the foreman that goes out and carries out protein synthesis. So how does DNA and RNA differ? I've talked about this before as well. RNA is single-stranded, whereas DNA is double-stranded. DNA is found in a helical form. RNA is not a helical molecule. RNA contains ribose sugar. DNA contains deoxyribose, and RNA contains uracil, while DNA has the thymine. There are 3 types of RNA. So we have the large, what everybody knows, ribosomal RNA, or the ribosome, has 2 subunits, a large and a small subunit. We have the mRNA, which is what is produced during transcription. Actually, all of these can be produced, but this is the one we typically talk about is mRNA, which carries the information of DNA in an RNA form. And then tRNA, transfer RNA, is what carries amino acids to the ribosome when you are producing proteins. Excuse me. So when you are going through protein synthesis, there are 2 basic phases, transcription and translation. So we'll talk about transcription first, then translation. Transcription is the production of the RNA from a DNA template. So remember, DNA is that CEO that never leaves the office, and so RNA are the workers. That's the foreman, and moves out and causes protein synthesis to occur. When I think about DNA, DNA is like a language, so it can't leave the nucleus. It's a language that basically your cells can't read, they can't use. So if you are transcribing something, you are writing it in a language that you understand, right? Or that other people understand. You'd have to understand both languages. So transcription produces a language that your cells can use. I always think about if you were, say, getting a you were reading directions, but they were in another language. You can't understand them, so you have to maybe use Google Translate to translate the information so you then can understand what's being asked of you. And then you can, once you have it transcribed, then you can understand what's being said. That's kind of what transcription is. We're changing the language from DNA to RNA, which our cells can read and understand. So DNA codes for the protein. Messenger RNA carries that code out into the cytoplasm where you can actually produce proteins. So each DNA triplicate corresponds to an mRNA codon. So we read our DNA. Here's our DNA. And we're going to produce, or we're going to use the template strand to make a copy of our coding strand of DNA. And so what I always like to show is that here, this is a template, and you have these complementary DNA or your RNA nucleotides attaching. If you look at the RNA nucleotides, they're identical to this strand, except for where you have T's, there are U's. So U, then there's A, G, C, A, G, C, G, A, and then U. Do you notice that? So this is the coding strand that actually codes for a protein. We use the template to make a messenger RNA molecule that is identical, except for we replace the thymines with uracils. Okay? And so again, here you have the RNA. This is from OpenStax again. I love my OpenStax textbook. I'm not going to lie. It's a wonderful textbook. That's what I use in my anatomy and physiology 1 and 2 classes. So here we have the DNA. This would be the template. This would be the coding strand. And here we're producing an RNA transcript of the DNA. And this is what I have produced in another class, but I use it to show. So the transcription of a prokaryotic organism. But here you can see I have the coding strand, and then you have the template strand. These are complementary to each other. If I'm using this if this is my coding strand, then I'm going to use my template to make my messenger RNA molecule. And so here I have the 3 nucleotide codes that are going to be complementary to this and correspond directly to this. They're going to be identical. So you have A, and then anywhere we have T's down here, we have U's here. Okay. And this is stuff that you don't have to know about, so don't worry about this because this is more for microbiology. But just know that when you transcribe DNA, you're going to use the template strand, and you're going to produce a complement of that template, which is going to be identical to the coding strand. Once you have that DNA transcribed into messenger RNA, we have to then use that information. We have to modify that information, so not use. We have to modify that information. So transcription of eukaryotic DNA produces an identical transcript of the DNA, but the majority of DNA doesn't actually code for proteins. So we have to cut out all the parts of the DNA that don't code for proteins. So in a DNA segment, we have regions called exons and regions called introns. Exons are the expressed portions. These parts code for a protein. Introns are parts of the DNA that don't code for proteins. And so they're interspersed within the DNA, but they don't code for the proteins. Make sense? So what we have to do when we produce that pre-mRNA or that immature mRNA, we have coded both the exons and the introns. We have to cut out all of these introns, which I always liken them to like commercials to when you're watching a movie. If you're watching a movie, the movie is focusing on something interesting. Commercials have nothing to do with the movie in general. And so we cut out those little introns or commercials, and then we splice together all of the exons and we produce a messenger RNA that has no introns. So this is a complete copy of just that protein. We then have to add a guanine cap and a poly A tail. So we've spliced out the introns. We have put all of the exons together, or we have cut out the introns and spliced together those exons. We then, oh, there we go. This is showing how this occurs. So we take intron and intron. We have exon 1, exon 2, and we just use a spliceosome to cut out that material, okay? We then add a guanine cap. A guanine cap is just a series of guanine nucleotides that we put at the front of the mRNA, and then we add a poly A tail at the end. The guanine cap and the poly A tail help to prevent digestion of the mRNA before it gets before it gets translated. Once we have that, then we can move into translation. And so translation is the production of the actual protein on the ribosome. I'm going to stop here because it's already been 12 minutes, and I don't like these to go too long because I figure, if you watch a video for 12 minutes, then you probably want to get up and walk around. Most people don't have a super long attention span. And so if you get up between each video, it's going to help you. So I'm going to stop here and I will pick up in the next video. Bye.